The brain cells are kind of isolated from the bloodstream by a barrier called blood-brain barrier. Let's discuss the histological structure of it. The first line are the endothelial cells lining the brain capillaries. There are no gaps among the cells, and the adjacent endothelial cells are connected with tight junctions. And second, it's a continuous type of endothelium without any gaps, without any fenestrations. The endothelium of the capillary is surrounded by a continuous layer of extracellular matrix called basal lamina. So altogether, this forms a so-called continuous capillaries. And that's the type of capillaries in the human body that has the lowest permeability at all. Moreover, there's another layer formed by the end feet of the astrocytes called pedicles. So the glial cells called astrocytes are sending processes that are embracing the surface of the brain capillaries. Let's complete one of these astrocytes. So this will be the cell body of the glial cell called astrocyte. And that will be the neuron here. With the nucleus and the cell body and the nissal substance, we can see that the neuron has not direct communication with the blood capillary. So these are the perivascular processes of astrocytes. This is the body of the astrocyte, either a fibrillar astrocyte in the white matter of CNS or the protoplasmic astrocyte in the gray matter. And this is the actual body of neuron. So the neurons are not exposed to all substances that are in the bloodstream, only to those that are able to pass that barrier. It's good to have in mind that the blood-brain barrier has not yet been formed or has not matured in prematurely born babies. Another barrier occurring in the central nervous system is the blood-cerebrospinal fluid barrier. The cerebrospinal fluid is produced in the choroid plexus which contains capillaries that are much more permeable because they are lined with so-called fenestrated endothelium. So there are microscopic gaps between and inside us the endothelial cells. So this is the lining of the capillary. This is the fenestrated capillary. Blood capillary. It's surrounded by loose connective tissue, so I will add some fibroblasts, fibrocytes perhaps, okay, some tissue matrix, and that's the loose connective tissue. With the capillaries, and the surface of the choroid plexus is covered by ependymal cells that are arranged as a simple cuboidal epithelium. So one layer of cuboidal cells ependymal cells they belong to the glia cells of central nervous system. They're arranged as simple cuboidal epithelium. Although by origin these are nerve cells, glia cells, not epithelial cells. Here is already the space of the brain ventricles. With the cerebrospinal fluid. And the cerebrospinal fluid is filtered through the wall of the capillaries, the connective tissue, and the ependymal cells into the brain ventricles. So this is actually a scheme of one of the viruses of the choroid plexus. And these are the layers of the blood-cerebrospinal fluid barrier, because here we have the blood and here the cerebrospinal fluid.